First, helicase unwinds the double helix and separates it into the leading and lagging strands. We've decided not to show the leading strand for the sake of simplicity, but in reality, the leading strand doesn't just disappear. Single-stranded binding proteins, or SSBPs, keep the two strands' hydrogen bonds from reforming. Primase lays down an RNA primer, a short RNA segment that gives DNA polymerase III, another enzyme that will appear later, a free 3' hydroxyl group to start building from. Going forward, it's important to know that the lagging strand runs in the 3' to 5' direction relative to the fork, anti-parallel to the leading strand. DNA polymerase III adds the complementary nucleotides in the 5' to 3' direction. Because the lagging strand template runs in the opposite direction, the lagging strand has to be built in short segments, called Okazaki fragments, each starting with a new RNA primer. The sliding clamp, this ring-shaped structure, locks DNA polymerase III onto the DNA, massively increasing its processivity. That means that it can add thousands of nucleotides without falling off, whereas without the clamp, it wouldn't be able to add nearly as many. This is crucial. As helicase continues unwinding the DNA, more single-stranded DNA becomes exposed, so more SSBPs fill the space to once again prevent the hydrogen bonds between the leading and lagging strands from reforming. Here, primase lays down a second RNA primer further down the lagging strand template, setting up the spot where the next Okazaki fragment will start. DNA polymerase III and the sliding clamp now synthesize the second Okazaki fragment, again in the 5' to 3' direction. In real DNA replication, the lagging strand is made as a continuous series of Okazaki fragments, one after the other, not in strict pairs. We're showing 2 fragments at a time simply to make the repeating cycle easier to visualize and because most textbooks will show it this way. DNA polymerase I arrives to remove the RNA primers and replace them with DNA by replacing the complementary RNA nucleotides with DNA nucleotides, moving in the 5' to 3' direction. DNA polymerase I uses the sliding clamp as well for the same purpose as DNA polymerase III. While it may seem like the process is complete, there are actually small gaps left behind between Okazaki fragments that need to be sealed. That's when DNA ligase shows up and seals the gap between the fragments, forming a continuous sugar-phosphate backbone. It does this by catalyzing the formation of the phosphodiester bond between the 5' phosphate and the 3' hydroxyl ends of 2 Okazaki fragments. These steps repeat over and over as helicase continues to split the strands and the replication fork moves, eventually producing a complete daughter strand made of many Okazaki fragments built separately and then joined together.